This is Jihan University's The Hague College of Pharmacy Medical Microbiology subject, Practical Lecture 3. This is 6th semester, of course, and I am Dr. Marwan Hussein. Today, our practical lecture is Identification of Staphylococcus on MSA agar as a selective media and by catalase test. Staphylococcus, of course, it is a positive bacteria, and it is pathogenic, of course, and it's a family, Staphylococcaceae, and contains more than 45, or today maybe recently is 50, more than 50 species under Staphylococcus genus. The main three species are Staph aureus and Staph epidermidis, Staph saprophyticus, and of course we have more than these three, but those are three major species. So the colonies on nutrient agar media is circular, convex, and smooth and creamy to colored to golden yellow in appearance, as you have seen in this slide. These bacteria produce and work in aerobic condition and do the aerobic respiration, of course. So they produce a variety of enzymes. One of the enzymes is catalase. So catalase is produced by many bacteria, but here in Staph aureus or in Staphylococcus, of course, genus, we can depend on this test to differentiate between Staphylococcus and Streptococcus. Now today we're going to explain for you how can we differentiate between Streptococcus and Staphylococcus according to this test. So the enzyme splits the hydrogen peroxide into water and oxygen. So hydrogen peroxide is produced by aerobic respiration, and it is lethal for bacteria if it is accumulated in the bacterial cell as well. So catalase degrades and breaks down the hydrogen peroxide in the bacterial cell before it can do any damage to bacterial cells. So bacteria like Staphylococcus produce this enzyme to break down the hydrogen peroxide before it can be any damage or injury to the bacterial cell. Of course, it gives me, as I said, the water and oxygen. So, as I said, it is one of the tests to differentiate between Streptococcus, which is negative in catalase test, do not produce the catalase, and Staphylococcus produce catalase, so it is positive. And we have two types of tests, of course. As I said, the Streptococcus do not produce this enzyme, so it is lack of this enzyme, and the Staphylococcus produce this enzyme, which is, as I said, we can do differentiate the aerobic, facultative aerobic, of course, of Staphylococcus. We have two types of test: slide test and tube test. So slide test, it's very easy to do it, of course. If I have the hydrogen peroxide, and I can get the microscopic slide and drop hydrogen peroxide on the slide test and have a one colony, small colony, or one colony by the loop, and then put in this drop of hydrogen peroxide, so will give me gas bubbles. So this is the gas, I mean it is oxygen, of course, formed immediately when I add this colony to the hydrogen peroxide. And the tube test, so look, this is the slide test, and this is the positive, and this is negative. In tube test, if I have in the hydrogen peroxide, 1 milli of hydrogen peroxide or 2 milli, and the I can have the absolute, I mean, hydrogen peroxide without any dilution, or I can have the 3% of hydrogen peroxide to the tube, and have a colony from the by loop from the slide, from the culturing of from media, I mean, and then put in the tube, which have hydrogen peroxides, and observe. You can observe and release the bubbles as you have seen in this slides. Look, this is the positive and this is the negative. So we're going to do this in the lab, of course. Just this, as I said, it's an introduction and to explain before I have and do it in the lab. On mannitol salt agar, the second section in our lab. So this selective media for Staphylococcus. This is a selective media of Staphylococcus, and we can depend on this media to differentiate between species of Staphylococcus and a selective media for Staph aureus. I'm now going to explain for you. If I inoculate Staphylococcus and Staphylococcus aureus or others on the mannitol salt agar, inoculate, and then incubate in the 37 centigrade for 24 hours, overnight, I mean, and then we will we can see that the reaction in this media. The principle is, so mannitol salt agar contains reagents and substrates and indicators, of course, which are the principle of the selective and differentiate media. Contains peptones and beef extracts, so a source of nitrogen, vitamins, and minerals, of course. Mannitol is a fermentable carbohydrate, so in their action the fermentation will happen and will produce the after fermentation acid production, and this acid production will be detected by phenyl red indicator. If the organisms, I mean like Staph aureus, can ferment mannitol, so an acidic byproduct is formed, and this will cause the phenyl red is an indicator in the agar will turn to yellow, from red to yellow. So this, I mean, it is positive for Staph aureus. Most pathogenic Staphylococcus, such as Staphylococcus aureus, most pathogen, I mean, Staphylococcus aureus 99% is positive. This is in general, of course. Will ferment mannitol and then the positive reaction in the media. The other Staphylococcus can grow in the media but cannot do this reaction, so will not turn the color, and just can grow in this media or selective media. I mean, the other Staphylococcus like Staph epidermidis and Staph phyticus or Staph lentus, Staph hominis, of course. Those are so can grow but cannot do this fermentation and change the color. Look, so this is Staph aureus, which is growth and fermentation and from red to yellow. Epidermidis, so can grow but cannot do fermentation. As well, saprophyticus can grow but cannot do fermentation, so those will remain a white colony. Look, this is positive Staph aureus and this is Staph aureus, but those are the other species of Staph, like epidermidis. Look, this is epidermidis, and Staph saprophyticus, of course. Again, this is the table which explains and shows the Staphylococcus aureus, the yellow colonies, the yellow zones. The Staph epidermidis, just colorless or red colonies with red zones. Streptococci, no growth, no growth and no any reaction. So no growth on the because Streptococci cannot grow on this media. Micrococci, again to large white to orange, and gram-negative cannot grow on this media. So this media, as I said, it's a selective media for Staphylococci species, especially Staph aureus, which do the fermentation. And the others, like Streptococci and gram-negative, cannot grow because it contains a 7.5% of salt in ACL salt. Our practical, and thank you for your listening.